Hello everyone. I'm here to discuss about tricarboxylic acid cycle. In short, it is called TC cycle. It has some other popular names called Krebs cycle, citric acid cycle, etc. Actually, it is the final common oxidative pathway for many of the nutrients we take, like carbohydrates, fats, and amino acids. Most of the energy, almost 70% of the total ATP produced in our body is through TCA cycle. Not only it provides energy, but also it provides many important intermediates required for the synthesis of non-essential amino acids, glucose, heme, etc. So try carboxylic acid cycle. Putting into a definition, TCA cycle is the metabolic pathway in which acetyl-CoA is oxidized into carbon dioxide and water along with production of energy. So the precursor for TCA cycle is acetyl-CoA, which is the uh, end product, or you can say the end product of glycolysis. Pyruvate is converted into acetyl-CoA, and this acetyl-CoA enters into TCA cycle. About 65 to 70 percentage of ATP is produced in TCA cycle. Already told. The complete cycle was proposed by Sir Hans Adolf Krebs. It was in 1937. So, in his honor, the cycle is also called Krebs cycle. Actually, he was awarded Nobel Prize for Physiology and Medicine in 1953 for this work. Hoxton and Potter identified the carboxylic acid as citric acid. So it is also called citric acid cycle. The enzymes for the pathway are located in mitochondrial matrix in close proximity to electron transport chain. This enables production of ATP without any hindrance. So it is to be remembered that TCA cycle operates inside mitochondria, and all the enzymes required for TCA cycle are located inside mitochondria in close proximity with respiratory chain or electron transport chain. It is an open cycle, which means that many molecules enter and leave the cycle at different stages, and we will see later. Thus, it is a connecting link between different metabolic pathways. It is an overview of a TCA cycle without the structures, of course. Pyruvate, which is the end product from glycolysis, which is converted into acetyl-CoA. Now, TCA cycle starts with a step combining acetyl-CoA with oxaloacetate, forming citrate. So this is the first step of TCA cycle. Acetyl-CoA combines with oxaloacetate to form citrate. So all the other reactions we'll see along with the structures of the respective compounds. So this is how we are going to start with TCA cycle. Pyruvate to acetyl-CoA. Now acetyl-CoA combines with oxaloacetate. It removes the coenzyme part, acetyl-CoA. You can see S-CoA. It is removed. The coenzyme A part is removed, and it combines with oxaloacetate to form a bigger molecule, that is citrate. This reaction needs water, and the reaction is catalyzed by the enzyme called citrate synthase. So this is the first step of TCA cycle. Now this citrate is converted into isocitrate. That means the OH group from second carbon, it is moved to the first carbon, and the enzyme which carry out the reaction is aconitase. In next step, with the isocitrate dehydrogenase enzyme, isocitrate is converted into alpha ketoglutarate. Isocitrate into alpha ketoglutarate. Now these hydrogens are removed by the coenzyme called NAD plus, which is converted into NADH and H plus. This NADH now enters into electron transport chain and it can generate 3 ATP molecules. Remember, NADH, which is a reduced coenzyme, it can enter into ETC and can produce 3 ATPs. Another change in the structure is a removal of the carbon dioxide, that is the carboxylic acid group from the second carbon. Here it is removed in the form of carbon dioxide. So the product is alpha ketoglutarate. Next step, by the action of alpha ketoglutarate dehydrogenase enzyme, alpha ketoglutarate is converted into succinyl CoA. You can see one coenzyme getting into the molecule. And again, the reaction is accompanied by reduction of NAD plus 2 NADH and H plus. You can see incorporation of this coenzyme along with the elimination of a carbon dioxide molecule. Next step, again it loses the coenzyme part by the action of succinate thiokinase, and the product formed is succinate. During that time, phosphorylation of GDP, guanosine diphosphate, occurs. It is phosphorylated to GTP, guanosine triphosphate. So there is phosphorylation of GDP to GTP. This GTP, again, it is responsible for the synthesis of 1 ATP. Succinate loses the hydrogen atoms and double bond will come between the two carbon atoms, and the carboxylic acid groups are oriented as away as possible. We call this molecule as fumarate. This is catalyzed by succinate dehydrogenase enzyme. Another coenzyme called FAD plus is reduced to FADH2. FAD reduced to FADH2. Again, as we have seen in case of NADH, FADH2 can also enter into ETC, but instead of 3 ATPs, it can give only 2 ATPs. So NADH can produce 3 ATPs. FADH2 can produce 2 ATPs. Now, a molecule of water is incorporated by the action of fumarase enzyme, and now fumarate is converted into malate. H2O. We can imagine as H and OH, H plus and OH minus. So the OH goes in this position and hydrogen is here. Now by action of malate dehydrogenase enzyme, it gives back oxaloacetate. Again, a molecule of NAD plus is reduced to NADH, which enters into ETC and produces 3 ATP molecules. So this is how TCA cycle works. Oxaloacetate combines with acetyl-CoA to give citrate. Citrate undergoes many transformation and finally it gives back oxaloacetate. So it's a cyclic process. The acetyl-CoA which enters the cycle is converted into carbon dioxide along with production of energy. Here, here, and here, and here. All these coenzymes enter into ETC to produce energy. Requirement of oxygen. Oxygen is not directly involved in TCA cycle. We didn't see any of the steps which takes oxygen directly, but the cycle proceeds only under aerobic conditions. This is because the coenzymes NAD plus and FAD plus can be regenerated in the respiratory chain or ETC only in presence of oxygen. I have told that this NADH, FADH2, etc. enters into ETC. When it enters into ETC, it causes the production of ATP and it comes back as NAD plus and FAD plus, which can again enter into another TCA cycle. So from TCA cycle to ETC, it goes in the form of NADH and FADH. It causes generation of ATP and comes back from ETC to TCA cycle in the form of NAD plus and FAD plus. So ETC works only in presence of oxygen. So all this process will take place only in presence of oxygen. The total reactions of TCA cycle may be summarized as acetyl-CoA plus 3 NAD plus FAD plus GDP plus a phosphate group plus water gives 2 molecules of carbon dioxide, coenzyme, 3 molecules of NADH2, 3 protons, 1 FADH2 and 1 GTP. Let's see the energetics of TCA cycle. How many ATP molecules are produced? Steps 4, 6 and 10, they produce the reduced coenzyme NADH. Each of them are responsible for a production of 3 ATPs. So 3 into 3 together 9. In step 7, a molecule of GTP which can produce ATP by substrate level phosphorylation. So it is responsible for 1 ATP. In step 8, FADH2. This will again cause generation of 2 ATPs. So total 9 plus 1 plus 2 equal to 12 ATPs. So the net number of ATPs produced through TCA cycle is 12 ATPs per 1 molecule of acetyl-CoA. I will see some significance of TCA cycle. As I mentioned earlier, it is the final oxidative pathway for all the major ingredients of foodstuff. See carbohydrates, broken down into simple sugars, and finally into glucose, converted into glucose. Glucose undergoes the process called glycolysis, and the final product, pyruvate, is converted into acetyl CoA. On the other hand, lipids, fats, etc., they break down to give fatty acids, and these fatty acids will undergo beta oxidation. The end product of beta oxidation is nothing but acetyl CoA. So all this acetyl CoA, they are funneled into TCA cycle for ATP synthesis. In case of amino acids, they undergo deamination reactions, which means removal of the amino group, and then it enters into TCA cycle at some point or the other. We'll see that. It acts as a source for reduced coenzymes essential for electron transport chain. Examples, NADH and FADH2. Its components can regulate many other pathways. For example, citrate inhibits phosphofructokinase, a key enzyme in glycolysis. Citrate can also activate acetyl CoA carboxylase, an enzyme which is involved in fatty acid synthesis. TCA cycle provides precursors for the synthesis of amino acids, fatty acids, proteins, and nucleotides. TCA cycle is amphibolic in nature because it is both anabolic and catabolic in nature. We have seen the first step, that is anabolic, because it is a building process. Oxaloacetate combines with acetyl CoA to give citrate. And again, we have seen breaking down of this citrate into alpha ketoglutarate, and alpha ketoglutarate into a smaller molecule, that is succinate CoA. So that is catabolism. It involves both anabolism and catabolism, that is building process as well as breaking down process. So it is called amphibolic in nature. The intermediates of TC cycle can be replenished or refilled by reactions called anaplerotic reactions or anaplerosis. See, many of the intermediates like succinate or oxaloacetate present in TCA cycle can be taken away for the synthesis of some other vital molecules. In such cases, the TCA cycle should not be stopped. For that, these molecules are supplied through other reactions. So such reactions which supplies the um, used intermediates of TCA cycle, that reactions are called anaplerotic reaction. These are some major anaplerotic reactions and contribution of TCA cycle towards metabolism of some other vital molecules. You can see some violet boxes. See, citrate can be used for the synthesis of fatty acids, sterols, alpha ketoglutarate. It is first converted into glutamate, an amino acid. Glutamate is again used for the synthesis of many other non-essential amino acids. GABA, which is a neurotransmitter present in brain, then purines, which are nucleotides, etc. Again, succinyl CoA, it is used for the synthesis of heme. Oxaloacetate is converted into another amino acid called aspartate, which is again used for the synthesis of pyrimidines, purines, and some non-essential amino acids. So all these violet boxes shows the usage of some of the intermediates for the synthesis of many other vital molecules. And I told that in such cases, when these intermediates are taken away, the cycle should not be stopped because we need energy. So we have some other side reactions which can refill these taken away components. See, all these amino acids, histidine, arginine, proline, they can be converted into glutamate, and glutamate can give alpha ketoglutarate. So if it goes like this out of the TCA cycle, it can be replenished or refilled like this. Again, when succinyl CoA is taken for the synthesis of heme, more succinyl CoA can be provided by some amino acids like valine, isoleucine, and methionine. So it is another anaplerotic reaction. Again, fumarate can be formed from phenylalanine or tyrosine. Oxaloacetate can be synthesized from pyruvate by the enzyme pyruvate carboxylase. So all these are the reactions, major anaplerotic reactions of TCA cycle. We will see the regulation of TCA cycle. The cellular demands of ATP are crucial in controlling the rate of TCA cycle. The regulation is brought about either by enzymes or by levels of ADP, which means that whenever we need more and more of energy, TCA cycle will run. So it is the cellular demand for ATP which controls the rate of TCA cycle. More the demand, more will be the rate of TCA cycle, and vice versa. Lesser the demand for ATP, slower will be the rate of TCA cycle. The regulation is brought about either by enzymes or by levels of ATP. Three enzymes are involved in regulation: citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. This is alpha. Citrate synthase. This enzyme is inhibited by ATP. That means when there are sufficient number of ATP molecules, there are plenty of ATP, we don't need to produce more ATP. So this ATP will inhibit citrate synthase. So TCA cycle will stop. So more energy is available, then TCA cycle will stop. Again, NADH, higher concentration of reduced coenzyme NADH as well as succinyl CoA will inhibit citrate synthase. Now the important enzyme is isocitrate dehydrogenase. It is activated by ADP and inhibited by ATP and NADH. So when there is more ADP, when there is more raw material, the process will take place and ADP is converted into ATP. If more ATP is produced, no need for running more and more of TCA cycle, so automatically TCA cycle will stop. Same with NADH. The third vital enzyme, that is alpha ketoglutarate dehydrogenase, is also inhibited by higher concentrations of NADH and succinyl CoA. Availability of ADP is very important for cycle to proceed. Unless sufficient ADPs are available, oxidation of NADH and FADH2 in ETC stops. Eventually, that will stop the energy production. Accumulation of these reduced coenzymes, that is NADH and FADH2, will inhibit the above mentioned enzymes. That's all about TCA cycle in brief. Thank you for listening. Thank you. Have a nice day.